Okay. Hello, everyone. Welcome to our virtual academic seminar to celebrate the launch of SmartMed. SmartMed is an open access journal co-launched by Valley and Tianjin University. Today is really our great honor to have Professor Ali Khademhosseini here to give us a seminar talk. So before that, please allow me to introduce him first. So Ali is currently the CEO and the founding director at the Terasaki Institute for Biomedical Innovation in Los Angeles. So previously, he was a professor of bioengineering, chemical engineering, and radiology at the University of California, Los Angeles. He joined UCLA as a Levi Light Child in November 2017 from Harvard University, while he was a professor at the Harvard Medical School and faculty at the Harvard MIT Division of Health Science and Technology. At Harvard, he directed the Biomaterials Innovation Research Center. He has published more than 650 papers with a current H index 139. That's amazing. So he is a fellow of a number of association organizations, including the American Institute of Medical and Biological Engineering, Biomedical Engineering Society, Royal Society of Chemistry, Biomaterials Science of Engineering, Materials Research Society, and also the fellow of the American Association for the Advancement of Science. So beyond research, Ali is also an associate editor of ACS Nano. This is a very prestigious journal as well. So today, it is really our good honor to have him here to share us a story entitled Engineering in Personalized Medicine. So without further ado, Ali, all floor is yours.
Thank you very much, Junchen, and it's a great honor to talk to you and the rest of the audience here. Obviously, I've always been a big fan of Wiley journals, and I think that the SmartMed is going to be a wonderful addition to the great family that you have. Just for people who don't know about a lot of the Wiley journals, I really think that the materials science journals are really some of the best. And I definitely think that the SmartMed is going to be one of the top journals as well. So I encourage everyone to apply, submit their papers, and cross our fingers to get acceptance. Also, I want to thank Professor Zhang for this great opportunity. It's wonderful to come and present across the Chinese family of researchers. And for people who may be wondering where I am, I'm actually here in Los Angeles, in an island near Los Angeles. And, you know, I came here to show you the nice scenery in Los Angeles, but now I have to compete for the sound again with the other people who are here. So wonderful to do that. So as Junchen mentioned, I have an experience that's been mostly in academia, including places like Harvard, MIT, and UCLA. But I also spent some time at Amazon, where I really wanted to learn more about the experience of being at a real company. And it's because of those experiences that I've recently started to work and really start this new institute called the Terasaki Institute for Biomedical Innovation. So what is the institute about is really, it's about trying to take the work that Professor Paul Terasaki did and build on it to create something really special. So what Professor Terasaki did is that he was the first person who developed the assay to be able to match donors and recipients for transplantation. So this obviously made transplantation a modern science and something that could affect the lives of millions of people. And based on this discovery, he also built a company. So he was both a professor and an entrepreneur, which is how he developed the impact of his research and some of the proceeds of the company became the basis of the foundation and the institute that we are pushing forward. So we're here in Los Angeles. As Jun Chen can attest, Los Angeles is a wonderful place. It's a beautiful city in the Southern California. And this is one of our buildings. We have 3 buildings at the Institute. This is our latest building. And what we do in this building is we basically do a lot of work on the following mission, which is to invent and to foster practical solutions. So you see both, there's the development of science here, but also the development of real world applications. How do we take the science and then take it into the real world through startups or licenses that are associated with different companies? And what we do is that our goal is to really work on personalized medicine in a number of different ways. So these are some of our buildings, as I mentioned. And to tell you a little bit about the kinds of research that we do work on a number of different personalized systems. We work on personalized biomaterials. These are materials that can go inside the body and understand about what the individual needs. And in many ways, they are smart. So they are smart materials that can interface with the patient and then be able to deliver the right kind of stimulations. There are also lots of devices that we are making that are personalized devices. These devices are basically the kinds of materials that we make to make systems that are basically have flexible electronics and sensors integrated into them. And we can take standard medical devices and make them better. We also work on personalized cells. These are things like stem cells or immune cells that have the ability to really generate a lot of personalized therapeutics and personalized cures in a way that we can tailor them also to an individual. Obviously, when you're dealing with cells, you have to make the cells that can be compatible with the person's own biology. We also work on other things like personalized implants. These are 3D printed medical devices or tissues that we can use for engineering, as well as this area of personalized physiological models, or what they call organs on a chip. For today, I'm going to mostly focus on these 2 areas, talking about some of our work on organs on a chip development and how material science and bioprinting can affect it, and also some of the work on how do we make 3D printing and 3D bioprinting technologies better by both developing new materials that can be better inks, as well as being able to actually make the 3D printers themselves better. So the way this is really done, these are such disparate, different types of technologies that the only way to really get them effective is to work with teams. And we worked on lots of different teams over the years. These are teams that are made from people who are engineers, biologists, and other things. And this is an example of the types of meetings that we have. As you can see in this picture, we have like biologists. We have even Professor Zheng Gu, who until recently was a faculty at UCLA and is now faculty in China. We have other types of bioengineers, clinicians, chemists, and many other disciplines that are working together. And we've always worked in big teams because we appreciate that there's a lot of diversity that everyone's thinking brings in. And we've always had a lot of people actually from China also come to our laboratory over the years, and they've made incredible contributions in pushing forward the science. We've been generously funded by many different entities, and through that, we've developed some of the work that I'm going to talk about. So let's talk about this area of personalized medicine, and specifically, I want us to focus on this area of personalized physiological models. So this area has become really popular because of the need that exists in drug discovery process. When you think about this drug development and discovery, what you see is that this is a very long process that takes many, many years, as well as it's a very expensive process that costs billions of dollars. So when you think about this process, you actually have a platform that is very, very it's like a funnel. You start off with many different things, and then you want to identify the one that is going to be the approved drug. So as you can see here, as you go through this process, you start basically spending a lot of money, and it takes a lot of years. Now, the process itself is made from a lot of in vitro tests and then things related to basically animal models and then different phases of clinical trials. Now, if you fail at a phase 3 clinical trial, as you can see, you've already spent hundreds of millions of dollars to be able to get this drug approved. And it's because of these types of failures that the R&D budget of pharma companies are really astronomical. And there's a lot of things that needs to be done to make better drugs happen. So what's the problem here? Well, the problem is when you think about why drugs fail, the main reason why they fail is because of an inability to predict their efficacy. That's how good they work, as well as their safety or toxicity. So this really is the major reason why drugs fail. But the other important thing is that even when a drug is approved, every single person behaves differently to this approved drug. So when you think about personalized medicine, you have to appreciate that people behave differently even to an approved drug. So understanding how this differences occurs is also very important. So when we think about a drug like Vioxx, for example, what we see is that this was a drug that was approved, and then it started behaving really bad and causing death in very, very low number of people, whereas it worked fine on others. So predicting this toxicity is very important. And because we couldn't do that, Vioxx had to be recalled and get off the market. So what's the problem here? Well, the issue is that our current systems where we do things like look at static cultures or current models like rodent models or mouse and rat models really don't mimic human physiology. We are very different in our behaviors within our body compared to what's in a dish and what's even in animals. So what's the solution? Well, the solution is, can we try to take the technologies that we've developed for tissue engineering and then be able to use these technologies for actually making systems that are better models of human tissues. So when you look at this, what you see is that bunch of different tissues that are made, and these are both barrier tissues like lung and gut and skin. They're metabolic tissues like liver, and they have other types of tissue functionality. And with all of these, the idea is to make the tissue as good as possible using human cells in a way that you can predict how a drug is going to affect human tissue behavior. So when you think about this process, what you see is basically many examples, some of which our group has developed, many of them our other groups have developed, and you start seeing trends. So virtually every tissue now has a model where people have taken cells from human and try to actually make the tissue model that is associated with a particular tissue and an organ. And when you think about these, you start seeing particular types of trends. You start seeing that basically these tissues have not just one cell type, they're made from multiple different cell types. They don't just have just a static culture. There's appreciation of the dynamic aspects of the tissue, like the biomechanics, how tissues change their shape because of how our bodies change their shape. There's, for example, like you see the lung tissue here, you see that the tissue actually gets stretched over every time we breathe. And this is mimicked here by being able to have a tissue that gets stretched. When you think about something like the blood vessel, you know, our inside our body, blood vessels have shear flow, which actually tells the cells what to do. So we can actually get this also working. And you see that there's a lot of appreciation of the architecture, the architecture of the tissues and the way different cell types come together. And we can take these types of models and then also be able to integrate all of these together to get better systems of how these things work. So that's the idea. We've been working on this area for a number of years. And one of the things that we've done is we started to build models that mimics this. And this is one of our early prototypes. Basically, what you see here is a lot of systems. These are things like fluidics and pneumatic pumps and valves, as well as a lot of different things like sensors and displays that can monitor what happens to the cells. Now, the main thing happens inside here. This is actually an incubator that houses the cells and the organs on a chip. And if you zoom into here, what you see are systems like this. Here you can see basically the platform. The platform is modular. That means that it's made from different components that we can combine in different ways, because many of the scientific questions that we ask requires us to be able to tune and model these in different systems. And one of the things that you can see here is that we can have different tissue models. We can actually have tissues that are made from different cells inside these chambers. We can have a lot of fluidic models. These are basically systems that can be fluidic models like bubble traps, pumps, and reservoirs, as well as having a lot of biosensors. These are sensors that are basically things like electrochemical sensors or physical chemical modules that can measure a lot of things about what cells are doing without actually destroying the cells and the biology that's associated with them. So this is this is schematic, and the systems actually, when you make them, they look like this. Now I put a few images here to show you the complexity of the microfluidics that we can make. We can make these microfluidics so that they actually work really effectively. So here in top, you can see a bubble remover. So you see a bubble come in and then it gets removed in the system. And lower on here, you see in real time how we can take these fluidics and change their channels and be able to change the fluids here to be able to move different liquids in here. So this is the system. And to make the tissues, what you can actually do is that you can make little microreactors. These microreactors basically have a little chamber that you can open and close. And inside these little chambers, you can basically put the cells that you want in different ways. So for example, you can put liver cells using techniques like 3D bioprinting, or you can use other techniques like light patterning to put liver cells in there. Or you can put heart cells in there using bioprinting or patterning using microstructured systems. So what I wanted to show you here is just two different tissue types, but there's many different tissue types that we and other people have been able to put and make using these systems. So I want to give you an example of the liver and the heart tissue that I just mentioned. So liver is a very important tissue for drug development, and it's basically made from a number of cells, hepatocytes being the primary ones. So human liver is very unique because it's actually doesn't really regenerate outside of the human tissue much. So it's very difficult to maintain human liver tissue outside of the person. Now, we've been able to do that by taking liver cells from primary sources. These are actual people's liver cells, and then combine it with other support cells like stellate cells, endothelial cells, and Kupffer cells to be able to basically combine these to make little tissues that have all the markers that you want and the enzymes that are associated with liver tissue function. And also they remain viable. These green cells here show live cells, and you can see that most cells are alive. And if you even track their behavior over long periods of time, they still remain viable. Now, what's also interesting is that you can take these liver cells and add drugs to them that were originally approved by FDA and then recalled because of liver toxicity. And what we can do is that as we start adding these compounds in different concentrations, we can start seeing toxicity associated with them. So they're actually predictive of things. We can also do things like try to do other types of tissues. And this is an example of the heart tissue. With heart tissue, obviously you can't take biopsies from a person because you need all the heart cells you can get. So what we do is that we use a technique called induced pluripotent stem cells. We can actually take cells from skin or blood and then be able to take the cells and differentiate them. First, make them into very primitive cells, and then differentiate them into cells that are basically heart cells. And by taking these heart cells and making microtissues, we can get microtissues that are functional heart tissue. They express markers that we want, and then they remain viable in dishes. They beat like heart tissue, and they beat very consistently, as you can see here. And more importantly, they have all the right microarchitecture that are associated with heart cells. And what you can do is that you can do the same kind of experiments that I mentioned for liver. You can add different compounds to it that are known to be toxic to the heart after they were already approved by the FDA and had to be recalled. And here, what you see is that, again, as you add these compounds, you start getting these more red cells, which shows that you're sensing the toxicity. But not only that, if you look at other things like the beating rates, what you do is that you can actually see the beating rates of these cells also drop off in a dose-dependent manner. As you start increasing the concentrations, all of a sudden beating rates go down. So those are healthy tissues. And I wanted to give you an example of a disease model. So one example of a disease model that you can make using these systems is progeria. Progeria is a very devastating disease. It's actually a disease that affects kids because of a genetic mutation. And it happens in this process. This is a process that is very important to the cell in the formation of envelope protein like lamin A. And what happens here is that as during this process, you have a post-translational cleavage that occurs. And because of this mutation, this cleavage cannot occur. And then you have the buildup of this protein that is not cleaved called progerin. Now, this progerin, what it does is that it actually causes a lot of inflammation in the cells because, you know, there's the immune cells start seeing something is not right and starts to some degree causing issues. So these issues cause slow growth as well as a lot of musculoskeletal and cardiovascular disease. So this is the problem, and we wanted to model this. And particularly, you know, why these kids die is because of their issues with their cardiovascular disease. So they actually get stiffening of the blood vessels that are basically due to inflammatory behavior of their smooth muscle cells. So we wanted to model this, and to do that, we took smooth muscle cells that are made using this IPS technology that I mentioned earlier. And we can take this smooth muscle cells from either healthy patients, healthy donors, or progeria patients, and then be able to make basically smooth muscle cell blood vessel models that mimic normal aging as well as this premature aging. So to do that, we wanted to make blood vessel-like structures that actually are functioning as well as possible. One way to do that is to actually make blood vessels and that can basically actuate. And these blood vessels, if you start looking into them, what you see is that they dilate. They actually change their dimension based on if you have a system that basically has this pumping of the heart. So if every time the heart pumps, the blood vessels actually change their shape. So we wanted to mimic this, and to do that, we made microfluidic channels that basically can deform and stretch every time there is a vacuum applied to this bottom chamber. And then the cells that are here, they start sensing this mechanical stretching that they would sense inside the body. So we can do that, and we can control the degree that these things actually are stretching based on the amount of vacuum that we apply, the force of the vacuum. And the cells can actually sense this. So this is actually the nuclear outline. You see that the cells actually change their shape of their nucleus. And we can actually use this now to start making models. So I'm going to show you one slide of the types of things that one can make. Here is basically an example of that. Here you have these smooth muscle cells that are from progeria patients. And we wanted to see how they behave in a Petri dish compared to how they behave in this microfluidic system that's biomechanically active. What you can see here are the expression of 4 genes. These are basically genes that are associated with inflammation. So if they're expressed, it basically mimics the disease, because in the disease, as you recall, I mentioned that the smooth muscle cells have a very inflammatory behavior. So when they're expressed here, then you see that the disease is actually being mimicked. So what you see here is basically the gene expression in a Petri dish is very low, but when you start putting them in these systems, when you have this biomechanically active systems, you have high expression of these genes. Now, what's interesting is that now this allows you to now test different drugs. And here's a couple of different drugs that we tested to see what their effects are in these tissue models. And what we see here is that basically we start seeing changes about how these different drugs affect the amount of inflammation that these smooth muscle cells are expressing. Now, we did this back in 2017, but what came out after our paper was results of a couple clinical trials, which basically showed that one of the drugs that we had predicted actually had a very positive effect. It decreased the mortality of these kids and decreased their cardiovascular issues and led to longer life. And this was really one of the first examples of how these organ-on-a-chip models can be used to predict and potentially help the drug discovery process, which is very, very important. Now, for the sake of time, I'm going to give you one other example quickly. This example is about a personalized medicine example. So imagine someone has cancer. Right now, what happens is that the oncologist can prescribe a number of different cancer drugs to that person. And some of those cancer drugs work and some of them don't work on that person. So one idea is to be able to take a biopsy of that tumor and then be able to take that biopsy and make a tumor model out of it that you can then actually test the drugs on. So here's an example of that. So we actually make a liver tumor model. And what we can do is that we can actually connect this liver tumor model to a healthy tissue of the same patient that can be derived using this iPS generated technique. So the reason we do that is because a lot of times when you have tumor drugs, cancer drugs, they actually have toxicity on the heart tissue. So we wanted to have that heart tissue present so that when we add drugs to test what kills the cancer, we can also see what the side effects are on the heart tissue. So this is the idea. And to do that, we basically made these organ-on-a-chip systems that are fully automatic. They sense a lot of the molecules that are secreted by the tissues, as well as they sense other types of things in a fully automated manner. So you just put this cells in and you let the reactors run, and then you can monitor what comes out of the reactors over time. And on top of that, what you can do is that you can now start building these types of systems in which you can basically add drugs like doxorubicin, which is a cancer drug, in different concentrations, and then see how it affects the tumor. So in this case, you want the albumin expression to go down because it shows that the liver cells that are tumor cells dying, and you want this marker GST alpha, which shows toxicity, to go up, and that's what happens over time as you start adding these drugs. And at the same time, you can look at the they say the side effect, the cardiotoxicity. So you can see that what happens as you start adding the drugs, what happens to this molecule, which is associated with heart toxicity, it goes up, and these compounds also cause beating rates and other things to go down. And you can monitor that, and potentially down the road, this could be important as a tool in personalized medicine to be able to identify which drugs are going to work on which people, and based on that, be able to generate data sets that can be used in machine learning AI algorithms to be able to predict these things without just by genetic information alone. So this is what I mentioned today is a very important area. It's whole area of using these types of tissues and material science to address a very important problem associated with personalized medicine and drug discovery. Now, I'm going to shift gears and talk about the technologies that are used to actually build these tissues. And a lot of these tissues actually require a lot of basically control over the microarchitecture. So over the years, we and other people have worked a lot on trying to control tissue architecture using a variety of different techniques. Traditionally, people have done this using porous scaffolds. These are degradable plastics or other type of gel scaffolds in which the cells kind of move around and form a three-dimensional tissues. When I first started my academic career, we were working a lot using microelectronics technology to be able to pattern surfaces or micromold materials to be able to generate structures with controlled shapes. Later on, we started to work on microfluidic systems, fiber knitting, and self-assembly techniques to be able to control tissue architecture and build complexity into our tissues. Now, more and more in the past few years, there's been a lot of work on bioprinting. That's basically the application of 3D printing to cells and biological tissues. And what you can see with bioprinting is that what it is that it's a technology that allows us to build tissue architecture in a very controlled way repeatedly, so that we can actually generate very controlled systems that can be used in these techniques like making tissues for transplantation or making tissues for drug discovery, etc. So what are the challenges that we want to address using these things? Well, we want to be able to generate complex tissues that basically mimic the architecture of real tissues. We want to be able to basically make these from multiple different cell types. We want to be able to have blood vessels in them so that we can make these tissues so that they become larger and they can have the vascularized tissues in there. We want to be able to keep the cells alive, particularly when we put them in the body. And before these blood vessels become functional, we want to maintain their viability. And also we want to make the bioprinters better. So these are some of the challenges that we have been addressing. For today, I'm just going to focus on a little bit of these, particularly on how to make the bioprinters better. So basically, for people who may not know, the printing process has been applied quite significantly to making tissue architectures. This is some of our early work where we could pattern and 3D print gels like agarose and then put the cells around them and then be able to get rid of these agarose structures so that you can actually generate these porous structures that you can vascularize. You can basically have cells in the gels around here, and you can even endothelialize them. You can put endothelialized cells that basically makes all of these functional. And really the big idea with 3D printing is to put the cells in the right architectures and allow the cells to reorganize themselves. So we initially put the cells in a particular gel or material, and the cells start identifying where they are, making the right connections with each other, and coordinating their development and maturation into more functional tissues over time. And as you can see, initially the cells are more rounded, but over time they start getting very tissue-like architectures. Now, I'm going to basically talk about some of the challenges that we've been addressing in material science of this, and also how to develop the printers and make them better. With respect to material science, we've been very much interested basically in making better inks that enhance cell function and survival. And with respect to printers, we want to make printers that are better, faster, higher resolution, and can also print with many different types of materials. So that's the logic with all of this. And to show you just some of the general bioinks that we've been working on. So these are some examples where you can see basically we have gels that can be used as inks. We can make other types of inks that can be used with virtually any type of printer. We can make conductive inks that can be used to make flexible electronics or conductively activated tissues like heart cells, muscle cells, and nerve cells. And we can make oxygen generating inks that can be used to keep cells alive after they've been implanted in the body. So I'm going to just give you one example of the type of printers that we make to address challenges. So when you think about making tissues, one of the things is that you want to be able to make tissues with different cell types, with different materials, in a very controllable way. So it's very important to be able to do that. And most 3D printing processes are actually designed for one type of material, and even bioprinters are typically designed for one type of cell. So a few years ago, we started to think about how do we make these systems better? Well, one way to do it is to actually get inspiration from nature itself. What nature does is that it has a number of different glands. Like for example, a spider has a number of different glands in its back and it can mix these the components of these different glands in different ways to be able to extrude the silk that it wants of a particular property. Now, spiders can do that, and as engineers, we can get this inspiration and actually come up with engineering systems that can mimic the same thing. So here you see 6 different channels. Each one has a different color, indicating a different material that can basically be used. And each one also has this valve that's computer controlled with pneumatic pumping. And by having this pneumatic control, you can basically control which of these materials gets extruded and can come out. So here, basically, you can have a lot of control over what comes out. So let's see how this is done. Imagine you have 3 different channels with 3 different materials shown with the different colors here. And you first open this channel that extrudes red material, then this channel that extrudes the green material, then the one that extrudes the blue material. Then so you can see that what comes out is a fiber that has all of these expressed sequentially. Now, if you open all of them at the same time, all of these will be expressed in the same region of the fiber that comes out. So by doing this, you can start having a lot of control over the kinds of things that you get generated. Now, the other interesting thing is that you can now get additional kind of control over this. So imagine the same 3 channels that I mentioned, but when you open them, you have regions when you're opening 2 valves simultaneously. So what happens here is that you don't only change the chemistry along the fiber, but you can also change the topography. Using other schemes, you can even have control over the surface structure. You can also have multi-phase systems. You can have systems that are basically have air bubbles, oil droplets, and cells embedded in these kinds of gels as well. So you can see that there's a lot of control over this that one can have. So we've been actually taking these and putting them upstream from printers too, which is allows us to now make these printers that can print different types of materials from the same nozzle, pretty much on demand, be able to extrude these in different layers, and even in the same layer, being able to print multiple different materials. So we've actually pushed this technology forward to make more advanced printers that can have, let's say, many different types of inks, and allows us to not only print these individually, but even be able to have combinations of these materials come out. So literally having thousands of materials come out of these systems from few different inks, kind of like how you make all the different colors in the world using just the 3 primary colors. This is the same way you can actually get lots of complexity coming out of these systems with the same kind of things. So these printers, the good thing about them is that you never have to change the nozzle to print different materials and cells. And it allows us to basically print on demand. It's much more effective than these printers that in which you probably have to change the nozzle every time you try to be able to change the material. And just to show you how this thing works, you can have different tissues that are made using these systems. And that basically it will allow you to build a lot of complexity in the engineered tissues that we're making. And these are all, of course, very early studies. But what it shows you is that the concepts can be used to actually make complexity in our tissues. And so far, for anyone who knows about 3D printing, they know that nozzle-based printers are just one type of printers, but there's a whole category of printers that are based on what they call stereolithography, that is patterning lights on surfaces and then be able to cross-link materials into those patterns. And we can actually apply the same principles that I mentioned to those types of materials too. So here you can see, for example, that there's a material here that can get printed on the surface, and then we can actually change this material using the same kind of multi-reservoir system that I mentioned. So you can initially have one type of material that you cross-link, and then be able to come up with a different material that comes out, and then you can actually cross-link that, and then be able to build multi-layer systems on top of each other and generate complexity in these types of stereolithographic systems. So this is all really allows us to now build complexity in these tissues that was not possible before. And this multi-material printing, I think, is something that is going to push the field forward and really going to help things like tissue engineering generate more complexity and even be able to print things not only outside of the body, but print things in the body, as we've been doing with some of our printers, and many other things. So with that, I just want to go back to the end of the what I said at the beginning about the concept of how engineering can help personalized and precision medicine. So when people think about this area of personalized medicine and precision medicine, people think about basically the application of bioinformatics to be able to take tumors and other samples and then be able to apply bioinformatics, proteomics, genomics to it to be able to identify the drug or the therapy that's going to work on a person. But we believe that there's a lot of additional engineering systems for this. We believe that one can make better tissues and implants using systems that I mentioned. There's a whole concept of making microphysiological tissues outside of the body, and also making materials that are personalized as well as devices. And all of these, when you think about it, they involve very heavily the concepts of making better materials, because all of these requires us to have these smart materials that are going to be functional and engineered to be able to respond to their environments. And that's why I think journal like a SmartMat is very important in the field, because it allows us to really address very important medical problems that requires a lot of personalization and tunability with our materials. With that, what I'll do is that I'll just tell you that the Institute is very much looking forward to hosting researchers. If anyone is interested, you can look at our website, terasaki.org. And I encourage people to apply. We always have a lot of people from all over the world, including many people from China, who come and do internships, and many of them wind up going back to China in very prestigious positions, which I'm very proud of. Also, you can follow us on social media. I just end the talk with this image because I think it's relevant for both SmartMat as well as some of the science that we're all doing. This is one of my favorite paintings by René Magritte, which is an artist in the back of the early part of last century. And what you can see is basically some of his surreal art, in which case he's looking at the egg and drawing the bird that's emerging from it. And I think that's really relevant because SmartMat, for example, is a very, at this point is still the egg, but I can see it becoming a very important journal in the field. And also a lot of the science that we do, both what I presented and probably what many of you guys do, it's still at this stage, but we have to really see the opportunity and imagine it and make it happen, because that's what we can do as researchers. So with that said, I want to again thank Professor Zhang and Jun Chen for this wonderful opportunity, and I'm happy to answer any questions in the remaining time that we have. Thank you.
Okay, thank you, Ali. Thank you very much for your wonderful talk. It's really amazing and also very inspiring. It's with a lot of eye-opening ideas and cool applications. Oh, that's amazing. So basically, I'm a messenger to pass some of question from the audience to you. So the first question from audience, that is, they want to know about multiple materials, extrusion, bioprinting, that's project published on Nature Materials. So people may think when you print in a different type of materials from and combine them together, so those different type of material components, they have different concentration, I mean, so they may diffuse to each other. So this type of printed materials stable, the fiber is not stable because the different layers of material may diffuse to each other, right?
Yeah, no, that's a really important question, and there's a few different things I can say about that. One of them is that it's actually in microsystems is diffusion and mixing is a very difficult thing, and diffusion is pretty much in many cases the only way of mixing, because you have these very low Reynolds number regimes. And also with the viscous materials, that makes it even sometimes more difficult. So our experience has been that it's actually more difficult to mix the materials than to not mix it. So for example, in some of our platforms where we're actually thinking about mixing different inks to be able to make something new, what we have to do is like play engineering tricks to induce the mixing and be able to do that. So in many cases, I think actually diffusion and mixing is the challenge. in these very small systems because of the inability to do convective mixing. So it hasn't been our problem yet. But I think that as we start also dealing with different things, there are challenges. For example, if you're having a material that's viscous and a material that's not viscous coming after each other, then there's challenges about how to be kind of make the extrusion continuous and uniform when you have materials with different types of properties like that. So there are challenges as you start changing materials. And those are things that one has to think about. I think if I had to kind of say what's the best way to apply the multi-material printing, I would say is that if you have one material that's printable, but then be able to add different cells or growth factors or other things to it in a way that doesn't change the properties of the material that's coming out significantly. So the viscosity is more or less the same, but the chemical kind of functionality, the biological functionality would be very different.
Yeah, well, that's great. Yeah. So here is another question that talking about the bumping rate. So when you use organoid, this kind of project, so you measure the bumping rate of the cells as with the relationships or the concentration. So you show the data 1 day or 2 days. So so how long for this kind of a cell can be in a living state?
Excellent question again. So I think, really the functionality of the tissues to a lot of degree depends on which tissue we're talking about. There are tissues that are more robust that you can maintain outside of the body much better, much more consistently. And there are tissues that are very, very, very sensitive to their local environment. Now, so, and really that changes, like, our tissues, we've been very much interested in maintaining them for at least 1 month because, to do a lot of this drug discovery, you need to do that. And to maintain the tissues for a month, you have to kind of think about many different things. One is that what are the cells that make tissues? Because a lot of times cells actually talk to each other. And some cells release some molecules to other cells and vice versa. So if we don't have those right kind of cell types, then you actually get much more lower behavior, a lot less survival over long periods. And then the other thing is to really try to appreciate a lot of this electrical, mechanical, and chemical cues that are in the environment of the cells. There's a lot of work that we and other people have done on that. But in general, I think 1 month is good for at least some of the more acute studies. For chronic studies, like if you want something that can have something that happens to our bodies over a few years, then I think we need to think about ways of accelerating some of these tissue models. For example, can we biologically speed up the process? Sometimes you can have older kind of patients in a way that they're more sensitive to particular types of things. So there are ways to address how long you want to keep them without actually keeping them very long, but be able to get information out of them as well.
Okay, thank you, Ali. Thank you for your answering with a lot of technical details. So here is a number of questions out there, but I just want to pick one last one. So as we know, you are the CEO of the Terasaki Institute of Biomedical Innovation in Los Angeles, and recently you have a lot of opening for PhD and postdoc student. So I'm wondering, so some of the candidates, so some of the people, some students wondering, so when you are hiring a PhD student or postdoc, so what's a personal characteristics you are looking at most? So when you select those candidates,
Yeah, well, that's a really important question, and I think I think that again, all of us do science because we love science and we love to make an impact in the world and try to do good, right? But But at the same time, science is a career. It's something that, each one of us who is in science also has to think about science and being able to do science long term. So I think, to be a good scientist, you need to have, of course, good scientific skills, right? You need to think in a good way. You need to be able to communicate effectively. You need to be able to be always kind of up on the literature and understand what's happening. What are the latest things that are moving forward? What are the fields that are important? Who are the players? And so a lot of scientific things. But I think there is additional thing on top of that. I think really good scientists also think about long term, not just short term. And long term means that sometimes you have to do the difficult things. You have to sacrifice. You have to, not take the easy path initially to make sure your long term is going to be effective. And I think in many cases postdocs, postdoc experience is like that. You do it for your own benefit. I don't see a postdoc as a job. I see it as a part of a journey of education, right? So I think postdocs who are very driven, who are passionate about what they want to do, who basically have both passion and perseverance, the ability to fail many times and continue to go at it until you succeed. I think those are the characteristics that are very important. And the other thing is learning every day, because if you don't improve every day, I think then that's a day that's wasted. So every day one has to think about, okay, how do I improve myself in a different way? And I think people who do that, every day they become better and better and better until they become really good by the time they have their own independent careers.
Okay, thank you very much. Thank you, Ali, for this kind of great suggestion and guidance to the young generations. So, okay, I think that's much of our talk today. And please take care.
Thank you very much. By the way, I just want to thank you guys once again, both Professor Zhang and Professor Chen, for this great opportunity. I am always I have so much great expectations for SmartMath, so I think it's going to be an incredible, and it's a wonderful to see it in good hands.
Okay. Hi, Ali. Hi, Ali. Thanks for your wonderful talk. Thank you very much. Thanks for your great support to our SmartMath, and also welcome to submit your work to SmartMath. Thank you.
Thank you. Will do.
Okay. Take care.